Hello, my name is Ben Farmer,
And my name is Melissa Harris.
and we are researchers with the School for Field Studies Center for Marine Resource Studies, or SFS-CMRS, located in the Turks and Caicos Islands in the Caribbean. Today we'll be presenting on ecological consequences of stony coral tissue loss disease in the Turks and Caicos Islands.
So the Caribbean is known as a coral disease hotspot. There are more than 30 known coral diseases in the region. Of recent prominence is stony coral tissue loss disease, which is marked by its rapid mortality, and it can kill corals within weeks to months. It is identified by rapid tissue loss of corals, revealing a blank white coral skeleton. So our hypotheses going into this research were that this disease would have negative and lasting ecological consequences, especially lowering coral density and coral cover. We also hypothesized that vast community composition changes would occur due to this disease, and especially due to the susceptibility spectrum, since some species of coral are very susceptible to the disease and others are not particularly susceptible.
So in order to survey the reefs of South Caicos, we looked at 3 sites: Plain, Spanish Chain, and Arch along the reef wall. So we looked at 10 and 20 meter depth, and we were trying to basically monitor the reefs long-term from 2012 through 2020. To look at reef health parameters, which were coral cover, richness, and Shannon index of diversity, we used multiple regression models. And to look at coral community composition, which is basically just the makeup of the reef, we used permanovas and NMDS plots. So what we found was that from 2012 through 2018, reef health parameters of coral cover, richness, and diversity were fairly stable. So they didn't really change much over that time period, as you can see in the bottom left there with coral cover. Things changed in 2020 as we predicted after the skittled outbreak of 2019. So coral cover and richness were at their lowest points of any year in 2020. Cover was significantly lower than any other year, and richness was significantly lower than most other years. Diversity, there was not much trend seen there from 2012 through 2020. There was a small effect of site and depth. So Arch had higher coral cover and higher richness than the other sites across all years, whereas 20 meter depth had a little bit lower diversity than 10 meter depth.
The community composition results showed that the composition of the reefs was significantly different in 2020 compared to both 2013 and 2014. So looking at the NMDS plot on the right, we can see that from 2012 all the way up to 2018, the years are clustered fairly closely together, whereas 2020 forms its own cluster off to the right. It's important to note for this plot, the way that it's created, the most variable factor is linked with the x-axis. So in this case, that seems to be years, with earlier years towards the left side of the plot and later years on the right side of the plot. We also see overlaid vectors, and those are of significant species as per a permutation significance test. Orbicella annularis and Diploria labyrinthiformis, those vectors point directly to the left, signifying that those species in particular were more prominent in earlier years as opposed to later years, like 2020. Site and depth also played some roles, as Arch was different, significantly different from Spanish Chain, and 10 and 20 meter depths were different, but these factors did not play as significant as a role as year did.
So in summary, coral cover on South Caicos is continuing to drop at a rate that, to our knowledge, is unprecedented, dropping to just over 1% in 2020 compared to 3% in the decade prior. Richness is also dropping significantly, and community composition overall is shifting substantially. So there's evidence on South Caicos that faster growing or weedy corals, such as Pérez estrioides, are going from an already fairly high abundance among the reef and the other coral taxa to an even higher level of relative abundance. So with Pérez estrioides, it is not known to be susceptible to skittled, so it makes sense that it's filling in the gaps. Whereas with Agaricea, another coral that is considered fast growing and weedy, we're actually seeing it decrease, which is likely due to the fact that it has medium susceptibility to the disease. The problem is that there's not large, important framework building corals filling in those gaps of agaricea. We're actually seeing evidence that some rarer corals like Diploria, as you see in the right-hand figure here, and Dendrogyra, those rare corals we're seeing them disappear from the reef. So the ecological consequences that we're finding here are comparable to those found in Florida in 2014, as well as in the Mexican Caribbean in 2018 when their respective disease outbreaks began.
So ultimately, this disease can have really big impacts on economies. So coral reefs in the TCI are estimated at about $47.3 million a year. And that is from tourism that the reefs bring in, as well as ecosystem services that these coral reefs provide. There are disease intervention methods that are going on in the Florida Keys and throughout the Caribbean, most recently in the TCI, to try to prevent the spread of this disease on individual corals. So this usually takes the place of amoxicillin pastes that are put on individual corals. Although this has been seen as effective on stopping the disease on individual corals, it doesn't prevent the spread from reef to reef or country to country. Ultimately, large-scale management will have to keep taking place to prevent this large-scale spread. Finally, this disease is not going to go away overnight, so it's important for us to keep in mind that these reefs that we dive on and research are likely to keep changing. We would like to acknowledge the School for Field Studies, as well as any SFS researchers that helped us along the way, all the way from 2012 till now, with in-field work and with CPCE analysis. And finally, we would like to thank the Center Dogs for keeping up our morale. Thank you.
Thank you.